Hi students, when we left off we were talking about cellular respiration. We saw the 3 steps of cellular respiration: glycolysis, the citric acid cycle, and the electron transport chain. I'd like to talk to you now about fermentation, which is something we've already touched on with our sauerkraut experiment. This is an alternate way that cells can break down carbohydrates to release ATP or create ATP. So this is a way that some cells can get energy from sugars. So fermentation happens in certain cells when oxygen is limited. Not all cells can do fermentation, and not all cells can do cellular respiration. Some can do both. So when oxygen is limited, we call that anaerobic. Aerobic means with oxygen, anaerobic is without oxygen. And there are 2 types of fermentation. Depending on the organism, an organism can undergo alcoholic fermentation or go through the process of alcoholic fermentation. This produces alcohol. This is done by several yeasts and some bacteria. The other type of fermentation is called lactic acid fermentation, and this occurs in many types of bacteria and can happen in animal cells. And when this happens, sugars are broken down to form lactic acid. So fermentation is a short process, not as long as cell respiration and not as complex, and it doesn't produce as much ATP either. For every glucose molecule that goes through fermentation, only 2 ATP molecules are created. So this can provide a rapid burst of ATP, but not a lot. So fermentation happens after pyruvates are formed. So after glycolysis, the pyruvates can either go into the mitochondria for aerobic respiration, or the pyruvates can stay in the cytoplasm for anaerobic metabolism. The pyruvates will be reduced by NAD+. NAD+ becomes NADH, and this will either form ethanol and carbon dioxide or the lactate I just mentioned. So with yeasts are single-celled microorganisms, and that picture on the right, just above my head, is a microscope image of yeast cells. Each one of those little capsules is a yeast organism. Some of them are budding, creating new organisms, and they reproduce through binary fission, making copies of themselves. And yeast will do that. They will reproduce when they are in the presence of sugars, and they'll make lots and lots of copies of themselves. If temperature is right and sugars are right, they're happy and will reproduce. And they will produce carbon dioxide, which will bubble up, and they will produce alcohol. And so this happens when we bake bread, actually, if you use yeast. You might mix the flour, which is starches, that is a polysaccharide, with water and salt and yeast. And once you make that dough, the yeast will start to divide the polysaccharides by hydrolysis, breaking them down into glucose. And then the yeast will absorb the glucose and produce carbon dioxide, and that gas causes the bread to rise. It'll also produce alcohol, which you probably won't taste because the alcohol will burn off in the oven when you bake your bread. This can also happen in the fermentation of alcoholic beverages. In the production of beer, for instance, you may make a mixture of maltose, which is a disaccharide, water, and yeast. And the yeast will cleave apart the maltose into 2 glucose monomers, absorb the glucose, and produce bubbly carbon dioxide and alcohol. So we use yeast to do this fermentation to make various products. We also use bacteria in the kitchen or in industrial food processing facilities to make other products. Bacteria can be used to make cheese or yogurt from milk by fermenting the sugars present in the milk into various substances. Or, like what we're doing at my kitchen, bacteria can be used to make sauerkraut. And there the bacteria will exploit and use the carbohydrates stored in the cabbage. And they will produce lactic acid, which is sour to the taste. A lot of people think that to make sauerkraut, you need to add vinegar, but that's not the case. As you saw in the video, sauerkraut only needs cabbage and salt. And then the water will come out from the cabbage, and it looks like vinegar at the end because it's a sour liquid, but it's actually a lactic acid solution created by the bacteria that were present on the cabbage leaves. So, fermentation allows for a rapid burst of ATP, and glycolysis can proceed fairly quickly, faster than oxygen can be obtained. So this can happen in animals when we are out of breath. I mentioned this in a previous video. If you happen to go running around the track or go for a nice long sprint, you will get out of breath, but your body still needs ATP to contract your muscles and go. Well, fermentation can happen that way, and our muscles will form lactic acid and go through that lactate fermentation. Lactic acid causes cramping of the muscles, temporary cramping, and so you'll feel that if you go for that sprint. The lactic acid will eventually be broken down and washed away, and you'll regain your breath, regain the oxygen to your cells, and you can go through cellular respiration, which is a lot less intense for us humans. So the chemical products of fermentation are carbon dioxide and ethanol, or lactic acid. And here's a picture showing some of the culinary products of fermentation. See those holes in the bread? They're from carbon dioxide. So fermentation is a lot less efficient at making ATP than cellular respiration. For each molecule of glucose, we only get 2 ATP molecules. And in cell respiration, for each molecule of glucose, we can get 36 or 38 ATP molecules. So this has to do with metabolism, because we've been talking about the catabolic side of metabolism so far. Catabolism is the breakdown side of metabolism. That's where foods or molecules get broken down into smaller units. So we'll take here energy-rich molecules like carbohydrates, fats, or proteins, and break them down for energy. Now, we've only been talking about glucose in cell respiration, but other carbohydrates can be used for energy as well. Polysaccharides can be cleaved apart through hydrolysis, and the resulting monosaccharides can be used in cell respiration, or other complex or simple carbohydrates even can be converted into glucose and proceed through the process, just like we talked about, the cell respiration process. Proteins can also enter in cell respiration. As proteins are broken down into amino acids, some of those amino acids are used to make other proteins. Excess amino acids can be deaminated in the liver, that is, having an amino group removed. This will result temporarily in poisonous ammonia, which will be converted to urea and excreted from the body. Some fragments of the amino acids can enter the respiratory pathways at various points. I'll show you a picture of that. So some amino acids resemble some of the intermediates already found in cell respiration and can be kind of absorbed into the process. Here we can see that carbohydrates are being respired in the cell respiration pathway down the middle. And on the left, proteins converted to amino acids can enter the pathway as pyruvates and be broken down and metabolized from that point on. They can also enter as acetyl-CoA or as an intermediate in the citric acid cycle. Fats also can be broken down into glycerol and fatty acids and enter the respiratory pathway at different points. This way, we can get energy from our food even if we don't eat any carbohydrates. Cells do easily respire carbohydrates, but you can respire proteins or fats. Now, metabolism isn't just the breakdown of reactants to products, but also the building up of molecules. So intermediates from the respiratory pathways can be used to build up different types of molecules. For example, carbohydrates can be put together through dehydration synthesis to make large starches, large polysaccharides such as glycogen. This is a storage molecule that we have in our muscle cells. Fats might be broken down or rebuilt to make adipose tissue. I hope you've enjoyed this presentation on cell respiration, fermentation, and the catabolic and anabolic sides of metabolism. Please read the chapters in your textbook and email me with any questions.